Welcome to the session of quality management in CDM. In today's session, we will have a brief introduction of how quality is managed in CDM. We will also understand the standards, the best practices around quality systems. Then we will understand something that's gaining a lot of importance in the industry these days, and that's called as risk-based quality management. It's just not risk-based monitoring, but also data management and the risk-based approach. We will then understand what are the different data errors, how should one identify those errors, the different kind of quality metrics, the inspection, understanding of the inspection, measuring the quality score, and finally, understanding the acceptable quality and error rate. Now, high quality research data provides the basis for conclusions regarding the safety and the efficacy of a medical treatment. So quality is a subjective term from which person or sector he's defining the quality. So in technical usage, quality will have two meanings. First is that the characteristic of the product or services is able to satisfy as stated, or it does as it is expected. And the second is a product which is free of any deficiencies, or a service that does not have any deficiencies, such as data management services provided and it has the best of the quality. So what is the term quality management? The quality management is the overall process, everything that's involved in establishing and ensuring the quality of the processes that are implied by the organization, the data that is being collected by that organization, the documentation that is associated with the clinical research activities. And therefore, quality management together encompasses both control of the quality as well as assurance or assuring of the quality activities. So the purpose of quality management is to provide the standard for design, conduct, performance, monitoring, auditing. So it gives us a standard structure, even recording, analysis, and reporting of the data. So for all of these actions of clinical trials, quality management is going to set a standard. It is going to set a bar. By doing that, by sticking close to the standard, we ensure that the data that is coming out of our system is therefore of appropriate, satisfactory quality. And it also ensures, by taking care of each and every activity for clinical trial, we also ensure that our patient's well-being is protected. His or her rights are protected during the entire conduct of the trial. So let's now understand quality assurance and quality control. Quality assurance is a set of activities which are intended to establish quality requirements and procedures. They ensure that the requirements that are met and procedures are being followed. That means, first of all, assurance is basically having all the required processes in place. Okay, and in order to have those processes in place, any of the requirements that are needed, they are also met and they are also being followed. Okay, now for example, in order to have good queries, it is important that the requirements is that we have good trainings in place. It is required that we have the right candidates with the right kind of uh, educational background. So those are those quality assurances in place. We have a right kind of a job description. We have the right protocol. We have the right written instructions, SOPs, and work instructions in place. So those are the requirements. So that is assuring that quality is going to be achieved. Then there is also a need to verify that the quality is being maintained. And this basically will include generation of certain procedural documents, which will guide the quality activities, and also the review of the documents to assess adherence to the written procedures, policies, and regulations. Okay, so to verify how quality is going to be maintained, we require a documentation of that the written processes, policies, and regulations. Then there is going to be tracking if our teams are trained. Hence, in organizations, you would often see that compliance towards SOPs and work instructions is being very closely monitored because that is the requirement of your quality. Apart from this, you would also have sometimes there would be regular trainings that will be given. And those self-assessment trainings would also involve some kind of tests. And it is a requirement that in order to clear the training, you need to clear those tests. Only then those trainings would be marked as completed. So these are some of the ways and means in which the quality is being assured, such that it is determined that the staff is trained on the policies, and only after they have completed the required assessments, they started working on the activities. There's usually also uh, an inspection, which basically checks if the individual was trained on the activity, and there should be a certification or a documentation that the individual was trained, and only after that the activity was started. So the start date of the activity should usually be after the certification date. So these are some of the ways in ensuring the documentation. Then in terms of certain site-specific examples, the site-specific examples would involve creating standard operating procedures, then a review of certain documentations, like there would be certain checklists that needs to be completed, and that will ensure that the quality process is being followed. So at the site, there's going to be a huge checklist which needs to be completed and shown as a documentation. It is a part of the trial master file. Then there will also be a review of logs and training logs, which will ensure that sufficient and relevant training has been completed and it has been documented. Then in terms of quality control is, again, how it is different from assurance. It's a set of activities which are intended to ensure that quality requirements are being met. So basically, it's like an ongoing review of an example would be review of the queries for data management team, or a review of the site visit report forms for the CRAs for the quality of site visit reports submitted by them. Likewise, on the site side, it will be basically audits and inspections, which will check if the data collection forms and records, they are completed well, they have the right logic. Then clinical study staff example here is completing checklist to confirm that each subject's informed consent process was followed correctly, or having a systematic comparison of EDC data with the medical records. So checking the CRF data against the medical records and seeing if things are being followed correctly or not. Also then from the essential document or the binders or the time trial master files, contents of the essential documents, and also documenting the results of the review. If the content is right, the TMF, all the required files have been placed, or is it that a specific milestone has passed, but that relevant documentation is still missing? An example of this would be there has been recent post-production changes. So a milestone was a post-production change, but relevant documents of post-production changes are not present. For example, all the changes, all the new versions of the CRF screens, they are absent, or the programming updates that were made. So the respective documents which the programmers and everybody has to sign in, all the respective stakeholders, before the database is put back into production. There is a move to production form, for example, and it is missing. So those are going to be your quality control checkpoints. By doing so, we ensure that before it was being worked upon, it had all the kind of relevant approvals in place. Let's now understand certain terminologies, what each of these different terminologies mean generally in the industry. The first one is the quality improvement. Now, we've already dealt with quality management, quality assurance, quality control. Okay. Quality improvement is systematic, data-guided activities. And as the name suggests, they are designed to bring immediate improvements in the overall activities that are being performed to deliver healthcare. Okay. So, and usually these improvements will be a result of your assurance and control. So quality assurance will have documented processes. Against the documented processes, control involves comparison of the actual processes and whether how close we are following it. Okay. When these quality assurance and quality controls do not match with each other, do not talk with each other, the result of that assurance and control process is quality improvement. You need to improve what is being lacking. Now, they're always for a quality improvement, and this is usually followed majorly for all these different organizations. At times, this has also taken a Lean Six Sigma approach for many organizations, especially if there is an activity that is affecting at the organization level. So there will be usually some kind of analysis and investigation involved for improving the quality. So first is you will be identifying the root cause of the problems. Why did the quality falter? What were the reasons for it? Then we will identify, after we have narrowed down on the root causes, the next step is to intervene and identify whether we can reduce the problems or we can completely eliminate the problem. What are the different possibilities? And then after we have identified what actions to be taken, then we take the actual process steps. We define those process steps. And usually these process steps, they are time bound, and they are usually assigned to certain people or roles who are immediately supposed to be working on those actions and close those actions. So identification, and mitigations, and then finally controlling the process. This is the usual way generally quality improvements are being worked upon. The identification of the solutions would generally involve whether there should be a change in the workflow or in time management of the activities. For example, if there is queries. So with respect to queries, whether it is the aging queries, the queries which are on hold, or the answered queries from the site which are on hold for a very long time, and that has resulted in significant impact because queries do not close on time, and the data update is not made in time in the database. So the data remains erroneous for a very long time. So how to hold on to this, or how to improve the quality of our queries? That could be a reason. There could be multiple reasons that could be identified as a part of this quality improvement process for on-hold queries. So the overall workflow, that is how when a system generates the query or when somebody generates a query within the system, it goes through how many people, or what is the usual process of escalating the queries which are kept on hold. That entire workflow can be looked at. How many people are involved? Okay, if it is the lead data manager to whom the data has to go, so for how many days will the lead data manager take to respond to those queries? If the lead data manager doesn't come back within that X number of days, who should be the next person who should be available to answer the queries, or who should it be escalated to? So such kind of escalation metrics, the workflow analysis will be done. Okay. It will also identify if there are any non-value add processes. For example, between lead data manager, there is one CDA, clinical data analyst, who is first given the queries. Now, generally across the studies, if it has been seen that the lead CDAs usually are unable to respond to such escalated queries, possibly remove the step completely and let all the queries directly go to lead data manager, for example. This will improve the overall time required for the data query to resolve. So that is one of the actions that can be taken from the workflow, assessing the workflow. Time management activities, that is how many days should be given to the lead data manager to resolve that query, should be usually 2 business days. If the current process says 5 business days, can we reduce it to 2 business days and still have the faster resolution of queries in place? So such could be an example of the identifying the solution. Then developing tools for source documentation. Okay. What are the current tools that are available to us for the source documentation? And are they proving effective? If not, can they be replaced? Or there can be an updations. Assessing the training needs. When we do the analysis, when we identify the root cause analysis, what is the issue basically? Why did the issue really result in a quality failure? So is it that the training needs were insufficient for the entire staff? Were there ambiguities in the way the training has been delivered? Or the training material itself is ambiguous at many places? So assess whether a retraining, any training needs are to be updated. Should there be more of practical hands-on for a difficult trials? Should there be more examples in the training material for the given processes? These could be various reasons that can be identified in the training needs. Then comes involving the appropriate staff in resolution. Now, obviously, if there is a quality issue that is affecting multiple studies, let's say for data management team, while doing the resolution at the organization level for the data management activity, it is critical that the staff involved are experienced data managers who have who have experience dealing with a lot of such different scenarios in different of their studies. They have seen enough, and they have also had lot of practice in identifying what could work best. They can envision which of the activities can be improved, and they can speak up those ideas. It will not be justified if we take a relatively inexperienced data manager to help resolve the identifying issues, because they would not have the relevant experience and maturity to give the solution to the problem. So involvement of the appropriate staff is important. If it's just not data management, but queries in general also affect the CRAs, or it also affects the medical monitoring team, then it is more appropriate that all the three roles, the experienced representatives from all the three roles, come together to make a common solution, which is more effective than individual piecemeal solutions. The different quality attributes for clinical research data. Okay. Now, there could be various quality attributes. What is the accuracy, the precision with which I work, okay, or the activities that are being performed? What is my accuracy level of performing the activity? Also, consistency. Can I produce the similar kind of results every time I do the activities? Or when the tool gives me a specific tool is being used to give me a reconciliation output or an error output, what are the chances that it will give me the same kind of efficiency every time? Okay. Then consumability. That is whatever work that is being created or the processes that are being laid down, how simple and easy it is for the staff to follow it to 100% compliance. Whether the process is current, is it relevant to do it? Granularity, do we have enough details on how to perform the activities? And lastly, there is no confusion around doing the activity. So these are the different quality attributes that are to be considered. And with experience and maturity, usually these are by default considered by any experienced, mature individual who will be laying down the quality guidelines for the organization. So for clinical research data, quality may therefore be referring to a data set that accurately represents the data points collected from the subject, has acceptable completeness, is defined sufficiently for use, is current, is attributable, and contains relevant data at the appropriate level of precision to answer studies, hypothesis, and the primary to answer the primary hypothesis of the study. So the study is going to be doing some kind of so all the trials are aimed to test a hypothesis related to the treatment. So all the activities that are performed during the trial are helping to assess that hypothesis successfully. Although the ultimate goal of a CDM personnel is to complete a study with a quality data set. Proper principles and practices must be employed throughout the course of the study to ultimately ensure the quality. If a study's design, protocol, case report forms are of insufficient quality, the study will likely or very unlikely will give you the accurate answers to the hypothesis of the study or the primary objectives of the study. So lack of quality processes in any part of a clinical study will lead to results that are distorted, they are missing or inaccurate. Let us now look at the industry minimum standards to assuring the data quality. The first one is to design and maintain data handling processes according to the organization's documented quality systems. That is, it is very important that there is a reason there are SOPs and work instructions, guidance documents created in an organization. And often personnel fail to oblige and work according to those relevant work instructions. At times, it's also been seen that people who move from one organization to the second organization would often just learn one way of doing things and they kind of repeat the same thing over and over in different organizations. Sometimes these may not be very as per the current organization's SOP and work instructions. So irrespective of whatever has been learned in one organization, it is important that the individual, before working on any of the processes, understands the current organization's SOP, understands the requirements, the kind of documentation needs, the kind of processes being followed in the current organization, and ensures that the activities are mimicking those or following or compliant with those requirements of the organization. Then, attempts should be made to collect only the data that are essential for the interpretation of the study results. And this is something that which has been a common mantra in almost all of our data management trainings, that the emphasis should always be on collecting the information which will be finally analyzed. Extra collection of information may not always be critical, may not always be advisable, because the efforts to clean that information may not really be required. So stick to the protocol, the actual collection requirements of the protocol. Then provide sufficient information in the data processing documentation to reproduce final analysis from the source data. There should be enough information that's available for the analysis. Assuring that the data quality for all studies, whether submitted for regulatory review or not, irrespective of whether we submit that information for regulatory. For example, for certain studies like observational studies, marketing studies, or for only publication studies, sometimes these will not necessarily go through the regulatory review. But irrespective of that, it is important that for every study, the data quality is assured. Then ensure that the data quality is appropriate for study analysis as per the parameters which are laid out in the statistical plan, if there is any one. Some of the organizations usually involve the statisticians right from the study start. So even if a statistical analysis plan is not yet created, the statisticians will provide their viewpoint in the setup of the study. So build that quality with the help of statistical purview right from the setup stage. Then use company as standards as much as possible for data collection and handling process, because using standards at any time is resource, time, cost effective. Also, there is consistency. Implementation of standards is easier across different studies. So, as much as possible, the first approach should always be to build standards. And if the standards are there, then the general options or the general process is to first look at those standards, adopt the standards, and only if required from the protocol perspective, then go for those study specific changes or updates. So here are certain best practices for assuring the quality. First is having an organizational policy, quality policy, which is strongly supported by the upper management, understood by the entire staff, and they are supported by all the operational procedures. So your processes are set up such that any kind of quality policy that is implemented can be easily adopted by the processes. Then maintain documentation of all the roles and responsibilities which are involved in managing a clinical study. This has gained so much of importance that you will notice that having a list of all the roles that were given access to the various study databases or different tools within the study, maintaining a log of accesses, constantly checking and revoking accesses once the roles are no longer working with the organization or with the study. This management is very critical throughout the phase of the study, and it's being regularly reviewed. So, access management of the staff, documentation of the staff, which includes their CV, their training certifications, their training dates, all of this is mentioned in, it's a part of the quality policies. And every organization will have certain rules and norms around maintaining this information. Then is using certain industry standard data collection and handling of the processes. So industry standardized, for example, CDISC standards, 21 CFR standards. Now these are standard guidelines. Now, obviously, because the regulatory team is also expecting data based on these guidelines, it makes more sense for any organization to adopt the standards which every other industry or which every other competitor in the industry is adopting. Using well-documented processes for data collection and handling. So every activity, when the steps are being performed, it has to be well documented, especially the data flow. When the data is being entered by the site, where is it entered first? From that central place, where else does the data flow? If the samples are collected at the site, the samples, how are they sent to the vendor? From the vendor, how does the data come to the internal team? All of these processes are documented through various different document names. And some of these we will be covering in the data management plan section. Then we have the minimize the number of data processing steps. to minimize the potential source of errors. So it is very evident as humans, the more complex we make steps, the more chances are to introduce certain accidental errors. So therefore, every attempt should be made to minimize the number of steps involved in performing an activity. Where also possible, to ensure quality and consistency of doing that activity similarly every time, it is best to also find out ways to automate the processes and have minimum human interventions, especially where the chances of introducing the errors may be very high. The next one is for the QA to identify any error preventions, and quality control processes should be in place to focus on the monitoring. So where quality assurance is all about having that milestone set or those guidelines set on how to prevent an error, quality control should also be inbuilt into the system to regularly check how compliant we are with our documented processes and compliance. So the final product, which is either the database that we release or any softwares that is going to be used in the clinical study, that should not be the focus of your quality assurance or your quality control. But then it's the process of getting that final product. Then ensure that the data quality, apologize, audits are being compared or are being done, and they are basically in place to assess compliance of the procedures to the regulations, compliance of the practices to the written documentations, conformance of the data to the source documentation, and conformance of the data to the written procedures. Apply the data to QC for each step of the data management. Apply data QC for each step of the data management processes. Okay. Quality checks, usually such kind on any data management process, is very effective. Lot of activity issues are being identified during this quality check process. Also ensure that all the data management people are trained on the organizational quality policies, and there is transcription available, training transcripts are available, which shows a documented proof that the individuals were trained on the activities before they started working on those activities. Let us now understand the different quality systems in CDM. A quality system in the CDM would comprise of the organizational structures, the different responsibilities, the different procedures, the processes, and finally the resources. These all are important to implement the quality management. Now, this approach was standardized by the International Organization for Standardization, ISO, and this is applicable across any industry, including the clinical research industry. A quality system approach will advocate an infrastructure that provides the flexibility to account for the differences in the studies in a controlled and consistent manner. Although not mandated, For all the clinical studies, a quality system approach has been adopted by the FDA in the medical device regulations. Every study should establish an appropriate minimum level of quality, which should be determined through planned analysis specified in the protocol or the statistical analysis plan. The assessment of the data quality needs should address the study's purpose, characteristics, and complexities. Now, within the context of CDM, a quality system should assure the following fundamentals. The first one is there should be written procedures in place and associated documentations that should enable the clinical database to be reproduced from the site's source documentation. Written procedures must be followed, such as your standard operating procedures, processes, guides, manuals, etc. Data are consistently of sufficient quality that support the conclusion identical to those drawn from an error-free data. That is, any kind of data anomalies, discrepancies are noted with relevant actions are taken as required. Now, let's look at the different components of the CDM that are taken into consideration. The first one is the processes. Defined processes necessitates that all the operations that are performed by the CDM are identified and they are defined. The starting point is an inventory of processes for which the department or group is responsible. The quality system standard also requires specification of the sequence of the process, as well as interaction between the processes. The quality system should be consistently applied to all the departments of the organization, because CDM is just one component of the clinical research process, but the data is going to be received by different other processes also. Like the same data is going to be reviewed by the CRAs team, and then there's going to be medical team, coding team, statistical team. So interactions between the processes, between the different groups is also important. For departmental implementation of the standard, document the interface points of CDM processes with processes from the other departments, something like a cross-functional process map that can be helpful. These interface points can be documented in even the SOPs for data management processes. The next one is the job description lists, and it describes the functions of the specific job titles. Position descriptions should accurately and thoroughly describe the requirements of a position, including the responsibilities, the tasks, and the education. Position description serves as the basis of candidate selection, training, performance, evaluations, as well as the promotions. Everyone involved with the study should have a position description that accurately describes the work they regularly perform, because often your job descriptions form an important part of your CV. And these job descriptions clearly identify what activities you all are trained on, what experience you have to have those activities to be done. And CVs are an important part of the trial master file, especially for the site staff. and the CRAs, as well as all the lead roles in the study. Next one is the oversight. Oversight from the management is generally a good practice. So even if a quality system will have documented work processes, there will be job descriptions given, there will be tasks identified and defined, trainings would be given, factors like comprehension of those activities, quality on doing those activities, the judgment and consistency, however, all of these factors will vary. Many of the CDM tasks will require review of the individual's work, as well as an opportunity for an individual to receive the constructive feedback too. So your quality management involves management oversight, wherein the managers would regularly feed back to the CDM personnel on the quality of the work that is being performed. If the quality is not as per the required standards, there will be certain remediations in place, and those remedial processes will be documented. Although there will be also some reviews which will be appropriately conducted by your peers. There will be peer reviews. Management oversight will occur above the level of the individual. An example of this would be departmental management will receive summary status reports for progress and QC of the activities. And even at a higher level, you will have management will have an oversight responsibility to see the assuring that the quality management system is consistently producing the acceptable quality. So there could be organizational level reviews that are being done. There will be project review meetings done at the project levels, which will talk about the different activities conducted, and there will usually be oversight managers assigned to those projects who will look at every activities and the personnel who are working on those activities. Then is the process control. The process control refers to the capability of a process to consistently produce a particular result. Although process controls can also be considered part of the management oversight, it is important to be described separately. Management is responsible for designing and maintaining the processes that produce consistent results. For CDM, consistent result may include acceptable database error rates, data timelines, minimal errors in database programming, and meeting milestone deadlines. Rather than establish separate measures and controls for each process control should be determined by the global organizational goals. High-level assessment may be sufficient, but there could be detailed measures on certain processes, which can help identify the issues early on. Training, both as per the ISO standard and the FDA regulations, both require that individuals have documented training for their job tasks. For each work process in which an individual participates, training will be provided and it has to be documented. Organizations usually also create a training matrix, and this training matrix is listing the position and the required trainings for that position. The job description should also be linked to the SOP, and it should be adequately represented in this training matrix. Here is an example of how the training matrix looks like. However, do remember that there is no regulatory requirement to provide these links. looking at the quality policy of an organization. Okay. Now, one thing is a quality policy for an organization is it's going to be at the highest level of the quality system. This is going to be specified by the top management. The quality policy will communicate and document an organization's intention with respect to quality. What is it that the organization is expecting? Hence, in most of the organization, you will find a quality uh, policy which is adopted at the organization level. This will have, so an organization should generally have a written quality policy. That's the expectation. And the top level management should demonstrate commitment to the quality policy by supporting the organization's infrastructure with adequate resources. There could be offline QC activities such as quality engineering, quality planning, procedures applicable to each study that will be enhanced by this infrastructure, and it will facilitate error prevention. Although an organization-wide quality policy is the best practice, if the quality policy does not exist, then data management should rely on the below quality guidelines or the documents like the data management plan, SOPs, any study specific procedures, and the study protocol in order to ensure that the quality is of extremely good is maintained. Because a quality product will never be achieved with only one department, adhering to a quality system, it is important for data management to elevate the need for a corporate quality policy to upper management and to inform the upper management of the working parameters that data management will apply in the absence of a corporate quality directive. Let us now understand about the quality manuals and plans. The quality manuals are defined by the ISO 9000 as a document which will specify the quality management system of an organization. An organization should have a written quality management, a quality manual that will define the quality practices, resources, and activities relevant to the data handling services of the organization. Most organizations already implement portions of the quality manuals as SOPs, but a quality manual is much broader. A quality manual should describe not only processes, but also training, management oversight, positions, and even the process controls. Quality manuals and quality plans, they should be flexible enough to address any differences in the various studies. For highly standardized organizations, information that would otherwise be part of a study-specific or project-specific plan, this could be included in the organization's quality system documentation. An example of this would be the quality manual, audit procedures, and SOPs. In these circumstances, the plan should reference these quality system manuals and detail how the document ensure data quality for each study. Quality plans may be designed to apply to one specific study or to all studies for which the organization takes full part or partial responsibility. The organization's quality plan or manual should also be subjected to versioning and change control. Then, what are the standard operating procedures? These are again a part of quality manual, and they are usually a large component of the quality manual. Organizations should have documented processes for creating, reviewing, and version control of the SOPs. To easily identify the time periods when an SOP should be used, effective date is usually assigned when the SOP version is published. Although they do not have to be archived with each study, SOPs should be available um, for the studied audit even after years of the study closure. Planned deviations from SOP should receive the same level of review and approval as the SOPs from which they are deviating. The level of standardization within the organization helps determine the level of detail that should be present in the organization's SOPs. For example, an organization with the standard CRF modules, database structure, and monitoring procedures may employ detailed SOPs and thus require less study-specific documentation. Study-specific procedures, a quality manual should account for the existence of any study-specific documentation. Each study have unique data handling needs due to variations in sample size, visit schedule, types of data collected, amount of data collected, and method of data collection. Organizations should clearly document study-specific procedures to ensure analysis database is reproducible from source documents. Study-specific procedures are also often known as the data handling plans, data management plans, data handling protocols, data quality management plans. Different organizations would use different names for these common plans. Such documentation should provide supporting details to the SOPs and will have a lower level of review and approval within the organization. Let us now understand audit, why it is important in a quality management for an organization. Now, the word audit is defined by ICH E6 as a systematic and independent examination of the trial-related activities and documents to determine whether the evaluated trial-related activities were conducted and the data were recorded, analyzed, and reported accurately as per the protocol, sponsor SOPs, good clinical practices, and the applicable regulatory requirements. So to summarize the statement, it basically is talking about an examination of all the activities that have been performed on the clinical trials, and it is site-related activities. Then once the data that comes into the different sponsor and CRO systems, how those were reviewed, they were recorded, how they were analyzed, all of those processes which are involved. All of this is then compared against the protocol, the operating processes or the SOPs and work instructions which the organization has followed while doing all those activities. It's compared against that. Then the regular good clinical practices, which is applicable to all the clinical trials across, and then the local applicable regulatory requirements as well. So, to be qualified as an auditor, okay, for CDM, one, the auditor should be aware or should have the knowledge of how to perform audits. That is, they should be aware of the auditing methodology. Then definitely, because these will be reviewing the CDM functions, they should be aware of how clinical data management processes usually are. Then certain basic computer programming fundamentals as well, so that they understand if there were any programmings that were being done in the system, how were these worked upon. Then also the industry regulations, that is, what are acceptable, non-acceptable regulatory norms, and if the data is compliant with those regulations. An auditor's training and experience will be sufficient to thoroughly and accurately assess the compliance of your processes with the good clinical practices. The audits of the CDM function should be performed very often to ensure that the CDM processes and QC procedures are effectively producing and consistently producing reliable, reproducible data for data analysis and regulatory review. So a comprehensive audit of CDM evaluates the entire CDM quality system. Now, following 3 levels should be examined in the CDM audit. The first one is the written processes. They should be compliant with the regulatory requirements and should specify process steps, decision points, which are required for handling and processing the clinical data. This will also include instructions for manual reviews, data entry conventions, and even the data clarification processes. Written procedures should be specific enough to enable the clinical database to be reproduced using the source documentation. To determine the level of compliance with regulatory requirements, an auditor will compare CDM processes with the current regulations. Then document the compliance of the organization or the department to the written policy, and that to the written policy of that specific organization. So compliance reports have to be presented to the auditors where it is evident that the organization was following their own policy. This evidence can be a part of, can include probably a database audit trail. It could have signed and dated checklists, signed and dated clarification forms from the site, and even interviews with the CDM personnel. The objective evidence should exist to indicate that the CDM processes resulted in a very quantifiable, high quality, reliable clinical data for analysis and regulatory review. Several steps are required to obtain objective evidence that CDM processes does produce reliable clinical data for analysis and regulatory review. The first step in quantifying the quality of clinical data, which is usually represented by an error rate. Additional objective evidence may include data demonstrating that an organization's data handling process is operating in a state of control Another type of important evidence is the assessment of the potential impact of the error rate on the interpretations of the data. That are ultimately derived from the data. Now, this type of assessment may be carried out by departments outside the CDM, but the results provided CDM with information that may ultimately improve the CDM processes. What are some other considerations for quality systems? Let's look at the EDC consideration. So, for studies which are using EDC systems, the data are available almost immediately after the data has been entered and submitted. And this includes the audit trail also available immediately, electronic signatures of the person doing the entry, as well as if there are any queries that fire. So review of real-time data is possible. And that allows the errors that can be identified very early in the system. At the same time, it will enable faster subsequent corrective actions also. Studies which are using EDC will also differ in regard to the source data verification. Okay. In terms of studies which are using EDC will have fewer source documents available for STV and as compared to a paper-based study, because most of the information would be directly fed into the EDC. There are certain quality considerations for regulated versus non-regulated market. So, regulated clinical studies will undergo additional scrutinies because the regulatory authorities would go into that clinical studies in greater detail. But even if this is not the case with non-regulated studies, the expectation is that the data quality is critical for any clinical study. The clinical protocol and analysis plan should drive the quality for any clinical study, whether they are regulated or not. One of the primary differences between regulated and non-regulated study is the level of the risk that is associated with the study. Due to the differences in the risks, the process employed and the degree of QC may vary between the two. An example would be that non-regulated observational studies will not need to go through as many frequent audits like as compared to a regulated study. And what are the considerations for external data sources? Vendors will be supplying data to the clinical study databases, and or the vendors will supply the data which will be integrated or added into the clinical study database, and analysis will occur on those vendor information. Hence, it's important that the vendor information is also of good quality. The recipient of the data must ascertain, usually through a vendor qualification audit. So there should be a vendor qualification audit that confirms that the vendor's quality system is acceptable and it will maintain the integrity of the clinical study database. A study protocol will determine what external data will be transferred into a clinical study database. This requires that CDM should be aware that the data is expected and communicate with the data provider to negotiate the details of the data transfer. If laboratory data is being handled by a central lab, communication will be on a one-to-one basis. If lab data is being handled by local labs, communication may be one to many bases, and that process may be more complex as compared to a central lab. The receipt of external data should be handled procedurally according to the quality system components, the SOPs, and the study specific requirements, which should be mentioned in the data management plan. Let's understand some minimum standards for data quality. Using statistically appropriate inspection sizes for measuring of the data quality. That means a sample size should be picked up which can effectively represent the entire population. Document the method and frequency of the data quality assessment in this data management plan or the quality plan. That means these documents should mention about the frequency of how quality assessments will be done. Perform at least one quality assessment of the study data before the final log, so that any of the data errors, they are picked up and remediation is in place before the database is locked. Document the quality findings and the corrective actions, so that at any time, if there is an audit, the auditors can be shown how the data quality errors was identified. And finally, determine the acceptable rates for the critical variables. Okay. In future slides, we will talk about what is the normal industry practice of accepting the acceptable error rates for critical data and non-critical data. But usually some companies have their own internal targets set for the acceptable error rates. So the companies usually have their own quality systems in place. They have their own QC or quality control assessments. The frequencies are being decided. There are even dedicated quality control teams who would regularly review the CDM data, the CDM processes, and will decide upon an error rate which the company sets as a benchmark. And often, employee goals are associated with those benchmarks. What are the industry practices on measuring the quality? The first one is using certain quantitative methods to measure the data quality. Quantitative methods for measuring data quality will involve classifying the data, then counting the data, and then constructing statistical models to help explain the database quality, database errors, and patterns of errors. Okay. An example of this would be for data management. Let's say it is the queries. So the number of queries that have been in a specific status, whether they are still pending with the sites, that is open status, or they are pending with the respective query owners, like the data management team, medical monitoring team, or the user who has raised the queries, or the site query rates. So there would be different classifications of the basis the queries. And through that, there will be certain statistical models which will help explain whether my database quality is at par. What is also the rate, the error rates. Plus, with this, you can also identify the pattern of errors. Is it that the specific site is issuing maximum number of queries, or has the maximum number of queries, and what could be the reasons for it? Database errors or findings can be generalized to the entire data set, and a comparison can also be made between the sample and the whole data population, as long as valid sampling and significant techniques are used. Compare the trial data and the process in the beginning, in the middle, and the end stages of the trial. Often, there are certain data snapshots that are created that are already built as a part of the study ongoing analysis. And these snapshots are at various milestones. When the first 50% data is in, then the next 75% data is in, and when once the 100% data is in. So by doing these snapshots, the study teams ensure that the data has been cleaned. Okay, so those can be defined. Usually, it's just not the DM team, but you can also have your clinical team who will work very closely with uh, the DM team to identify uh, or predefine the criteria to trigger the site comparisons, right? And based on the monitoring reports, we can also perform some quality control on 100%. Okay, so perform quality control on 100% of the key clinical safety and efficacy variables. Now, obviously, because these decide your primary and secondary endpoints. Okay, so if they decide the primary and secondary endpoints, it makes more sense that we perform 100% quality check on the data that is being submitted, because that decides the phase of where exactly how good the trial is being conducted. Also, monitor the aggregate data by site to detect sites which differ significantly. So a lot of times good uh, trends are identified when the data is looked at as a whole, and there can be appropriate corrective actions that can be taken. An example would be if looking at all the data processes, the data that has been given by a specific site, there could be trends that can be drawn. For example, if there is over recruitment of the site and the number of AEs that are being reported, they are constantly, there is very little variations in the AEs. So the recruitment is high. The number of AEs being reported is also consistently high, but there is very little variations between each of those AEs. The number of queries are also high for the site. Then definitely this is drawing a pattern for that specific site, that the site is recruiting but is letting go on many compliance aspects, or the data is not reported truly as it should be, and there could be certain data flaws. So when such trends are identified through these QC processes, quality checks can be controlled, the sites can be immediately controlled, there could be increased monitor on-site activities can be performed, a detailed review of each data that has been updated by the site that can be specifically done. And if the norms continue, if the issues continue with the site, there can be a common decision taken with the sponsor on the future of the site with the study, so as to not to impact the data quality for the final submission. Lastly, we have to perform one quality control prior to the data release for decision making as to what kind of level of cleaning is required. Okay. At times, these also change the course of the trial, especially the adaptive trials. Now, basis uh, the how the data has been received by in the database, and what course of actions to take, where exactly the data is pointing, what kind of changes should be made into the process or the protocol. All these decisions are being done. Therefore, quality control is required. A QC of the data is required. Let's now understand risk-based quality management system. Now, this is basically talking about identifying where exactly your data is at risk and then concentrating your efforts towards mitigation of those risks. So the entire system is now built on identifying where errors can most likely happen. So there was a market research and it determined that even though SDV consumed a very large amount of time, so most of the monitoring visits, there was a significant part of that was being taken by this SDV activity. And what is SDV? SDV is basically comparison of your source data with the CRF data and just checking if source data information was copied correctly into the CRF. So identifying basically just the transcription errors. And it was realized that while doing this, the total percentage of change because of that SDV is just 1.1%. So the amount of effort required was way too high than the result of just 1% data update. And obviously, it's just not time consuming, but it is also increasing the overall cost of the study. So with this knowledge, sponsors of CROs are now exploring targeted SDV approaches, which means that the SDV is going to be directed more towards the site which has performance issues or where the data criticalities are way too high, which means that if a site is doing well, centrally, the central data monitoring team doesn't identify a lot of issues in the data that is being reported by the site. And other regular monitoring activities also do not result in a lot of data issues. Such sites will have a little lesser SDV approach as compared to a site where there have been a lot of issues identified, or the site is very new to the entire clinical research activity and they are still a novice. They are learning the ways of handling data, handling clinical trials data at their sites. Or the sites are historically not very high performers in providing good quality data. So your SDV would increase on such sites where the risk of data errors could be more, rather than the site who is relatively well professional, they understand how to handle their information and handle the clinical data. So what is going to basically happen is the SDV for critical data and the site with less performance will be more concentrated, and there will be less SDV cadence for sites that are performing well. Okay. Quality management in site monitoring will also involve something called as the source data review. Now, source data review is basically a review of the source documents with respect to the protocol. That is, now they are checking the quality of the source data here, whether the data is collected correctly as per the protocol or are there deviations. So that means source data will have much more information and not every information in the source document may be recorded in the EDC. Okay, so this is just the verification that as per the protocol, the data is getting collected properly, the activities at the site, the activities at the site, that is being conducted well, the assessments are performed correctly, which is showcased by source data getting updated correctly. So centralized monitoring is usually the ones who support in this risk-based quality, wherein this team will be centrally located, and they are usually either sponsor central monitoring team or the CROs who are handling the clinical trial on behalf of the sponsor. So this team will be monitoring the site data very closely, and they use a lot of analytical tools. They'll use a lot of visualization dashboards. And through this, they constantly keep looking at the data, once it is being fed by the site. And that aids the clinical operations team to determine the site performance and thereby target the SDV site monitoring to difficult sites. So the industry is now exploring quality by design, that is including or putting the quality right in the setup stage. So when it is there in the setup stages, there are minimum quality issues to begin with. So they are looking at how quality can be installed right from the study protocol draft process, okay, as well as all the initial setup processes that are being used. Focus is on critical to quality factors. That is on the critical parameters. The focus is now moved to this rather than every data. And to ensure that the protection of the study subjects is there and there is reliability on the data that is collected. The correct management of the risks which are related to the critical quality factors. That is, they're basically shifting their focus on what data truly matters. So what are data errors? So a data error, since we've been talking so much about concentrating our efforts to data errors, this is defined as a data point which is not giving me the right value. The data point has an incorrect represented value. A clinical research study will be a very complex process. Okay, some of the protocols with multiple arms, complex, multiple investigational products, many different kind of assessments to be performed. This makes the project very complex and it involves many processing steps. So a discrepancy between the data set and the CRF that cannot be explained by the data handling conventions or by any site signed clarification forms. Okay. So there's this final data set which is being reviewed by your stats team. And why is there a difference between what is coming in the data set versus what is there in the CRF? And if there is no explanation why what is missing from the data set, either by certain standard data handling conventions, that is a standard data handling document which has listed down the reasons why you see a discrepancy, or in terms of paper CRF studies, the site hasn't sent any clarification forms explaining why the deviation is observed in the data, or any programming conventions also, which is not present. Okay, and this programming convention is again defined during the trial analysis period. So when the discrepancy cannot be explained, that is data error. Each step where data are transcribed, transferred, or otherwise processed has an error potential associated with it. Okay. A data is defined as a data point that inaccurately represents a true value, like we said. So there are many sources or causes of data errors, including but not limited to: A, site has done a transcription error. Other one is data processing steps were incorrect and that led to an accidental missing the errors. So data processing did not identify the output, they did not identify the data errors. Intended responses because of ambiguous questions. So if the CRF designs was not right and the questions or the data points on the CRF design was confusing in such a way that the responses were not correct. The responses were not the actual responses which were required as per the actual events that occurred at the site. Again, that would lead to certain data which may not be a true picture. Or there is a collection of the data which is outside the required time window. So there are many deviations in the way data has been collected. So what are the error detections? It is not practical, not even necessary, or even is efficient to design a quality check for every possible error, which is not possible. Also, it is not possible to perform 100% manual review because manual review itself is very accidental, is prone to a lot of misouts. So there will always be errors that may not be addressed by the quality checks or reviews, and there will be error slips that go through the quality check process undetected. So in spite of QC, there could be chances where errors may slip. Okay. Program checks, like your data validation checks, your listings, and your edit checks. Now, they should be applied constantly, consistently across the trial data. And all the errors that are identified in this manner can be corrected. At a minimum, these checks should target your critical fields. Ensure that at least at the critical fields, program checks are present. So that errors which will have the greater impact on the outcome of the study, though, can be at least arrested. However, it is a possibility that not all the errors can be detected using this method. An example would be unreported AEs. Since nothing is reported in the CRF, there is no way for any data validation team to ascertain if AEs are truly not occurring for a subject or are they being underreported for a subject. Or there could even be another issues that programmatic checks cannot be programmed for certain text fields. Remember from the CRF design training, one aspect was very clearly said that if the text fields should be minimum because analysis of the text field becomes very difficult. What goes into the text field cannot be really determined, it cannot be predicted. Hence, there cannot be any checks that can be run on those SAE narratives field, let's say. So here are some of the detection methods that is present, that is being given by the SCDM and in the GCDMP. The source data verification, as we say, source data verification. This could be used to identify errors which are difficult to catch with programmatic checks. An example would be if the medical records are not matching with the CONMED or the AE form, logs form of the CRF. Obviously, programmatic checks do not know this. So only an SDV, a source data verification, will be able to identify those errors. Any discrepancy between the two that cannot be explained by the CRF completion guidelines, the protocol, or the approved site conventions, these will be counted as errors. In addition, if the study is using EDC methods, okay, SDV may be the best way to check the data errors. The scope of the SDV is usually decided on a trial to trial basis and should be determined at the beginning of the trial. For example, now, like we said in the previous slide, targeted SDV. Now here, what basically is being done is, when we say targeted SDV, it could either be that we are looking only 100% SDV of the critical forms of the entire study, which means that only that specific critical form for all the subjects in a given study will be SDV'd by the monitor. Or there could be a second approach of targeted SDV where 10% of random subjects will be picked up from the total subject population, and there will be 100% SDV of those 10 subjects. So depending upon the trial need, targeted SDVs can be determined to identify any errors. So as you can see, different detection methods. We have programmatic data checks, which are usually your edit checks and listings. Data validations is again through listings that are being created. Reconciliations by the data management team. Then there is something called as aggregate statistics. So this is usually done by the statistical team, and then they identify data trends, and based on data trends, they usually share those findings with the study team, who then investigate with the site if the data that has been reported is correct. Then, which are the quality metrics? Startup metric is to measure the efficiency in the quality receipt of the regulatory packages which are approved by different countries. Quality score of protocol versions before final approval, site selection score, how many sites were not selected during the SSV, that is the site selection visits. These are some of the matrices which talks about the overall quality process. So in the startup metric, we have, like we said, the regulatory packets that were approved just in the first receipt. That is, the countries are not re-asking for certain documents. This shows about the preparedness of the team in providing the right documents in the very first go. Then quality score of the protocol. How effectively is our site selections happening? How many rejections, percentage of sites which are not getting selected during the site selection visit? Then the number of versions of prior to the protocol approval. How many times were the drafts updated? So those are some of the good kind of metrics in the startup. In the conduct phase, again, from the data management perspective, there could be the number of queries and the requeries, aging of those queries. That is how many days they were present from open to answer, which means that once site answered the queries, how much time did it take for the query owners to close or action those queries? Then answered to closed status. How much time did they take to close the queries? How much time did the site take from change in answering the queries? So that shows, it becomes a site metric, how efficient your site is. Then there will be metrics which are generated from the audit trails. Your audit trail is going to tell how many times the site is regularly modifying the information. Also, sometimes trends can be identified, whether the site is regularly going and updating the information. And then when the query fires for the data non-compliance, they change it back to compliant data. And as soon as the query is closed, they again go back and update the data to the original value. So such kind of data issues can also be identified. How many times a data item has been deleted. Like the example I said, data is being changed after central team verifies that the data is now correct. After that, there are changes made into the data changes are being made for the same subject information. And also data entry lag. Data entry lag is basically if the visit occurred, after how many days of this visit occurrence did the data enter into EDC? The expectation is that our EDC data should be real-time data. That is, once the activities are performed or the assessments are performed at the site, the expectation is that within 24 hours or within the required data, as soon as possible, the data has to be entered into the EDC. But there could be, through audit trail metrics, you would realize that there's some data entry lag. Some sites do it within 5 days. Some sites are doing it within 21 days. So after 21 days of the visit, the data is being entered. So such kind of things can also be created as a metric, an indication of the quality of the sites. Then is closeout visits. For a closeout phase of the study, what are the different kind of protocol deviations that have occurred? Are there any kind of protocol deviations? Then what were the data errors? The number of data errors. So in the final data sets, when the analysis is being done or the review is being done, we identify the number of errors to the total number of fields that were reviewed by the quality reviewer. So that could be some of the matrices. So another important aspect of quality is inspection, and the E6 or the Good Clinical Practices GCP that defines ICH inspection as the act which is done by usually the regulatory authorities, and they conduct an official review of everything. That's the documents, the facilities, recordings, or the records, or any other resources which the authorities will feel are related to the clinical trial. So anything that is related to the clinical trial will be officially reviewed by these authorities. And either they could be at the site, or it could be the sponsor facility or the CRO facility, or any other establishment, even the vendors at times, as which is appropriately felt by the regulatory authorities. The American Society for Quality, which is also defines inspection as measuring, examining, testing, engaging one or more characteristics of a product or service, and then comparing the results with the specified requirements so as to determine whether conformity is achieved for each of those different parameters. So now The term inspection is used to indicate a scope which is narrower than a comparison. And it's a process where measuring can be performed as a step in the work process with less independence than comparison. Okay, so inspection is a narrower process. For example, a CRF to database inspection. This could be performed by individuals in the same department also, because comparison is happening, or the same project team as the one who did the work, as long as they are not the individuals who perform the work being inspected. So any peer review can become an inspection. In contrast, what is comparison is often performed by trained company or sponsor representatives. So you'll have specific people coming for it. And many organizations will require both ongoing inspections or more formal comparisons to assure high quality data for all their trials. So data comparison is basically that errors can be detected by comparing two representations of the data, which are captured at different points in the data handling process. A CRF to database comparison is performed by comparing the CRF to the data stored in the database. Depending on the needs of the study, the comparison may be performed on clinical database immediately following the data entry, or it could be even done on the analysis data sets at the database log. In either case, an error is defined as a discrepancy between the data set and the CRF that is not explained by data handling conventions, site-signed data clarification forms, or programmed conventions defined in the trial analysis plan. Next is what is sample size then. Usually, sample size should be a good representation of the entire population. So quality checks will be performed on the sample size. And the best way to determine a sample size, or the best practice across the industry, is to come to a sample size through statistical selection, or through statistically determine a sample size for any kind of inspection. By doing this, you will ensure that the information that is obtained from the inspection, that is a fair representation of the entire database, because your sample size was a good statistical representation of the entire database. And therefore, that can be used in decision making, the results of the quality. It is important that the data manager will work with the key study personnel to develop and document a sampling methodology. Okay, and usually it's well documented how the samples should be collected. Now, for the general practice around across is that for studies which are having very large population, common sample size algorithm is square root plus 1 of the total population. Okay, so square root the population, total subject population, and then plus 1 to the result. That becomes your sample size. Another approach is also, which is frequently also used across the industry, is using a 10% sample of the total subject population. So what are the error rates? Data quality can be quantified in two ways. First is the number of rows with the errors in the data set. So you have one data set. So number of rows that had errors in the data set. Another way to do it is doing through error rate. Error rate is more preferable And we will see that in an example below. Error rate is also more preferred because it's easy to then even compare the quality across tables, across different trials also, across different data sets, and it avoids any misinterpretation of the error counts. Okay. So error count of large data set versus error count in the smaller data set. Okay. So how this is being done, let's look at this here. So the error rate is defined as total count of errors divided by the total fields that were inspected. So from the total fields, how many errors were there? When you divide it, you would get the error rate. Now, error rates are sometimes expressed as the number of errors per 10,000 fields. So you can simply extrapolate this in terms of whatever was your current count. So if this is the error rate in the current count of the fields, what will be the error rate when the count of the fields is 10,000? Okay. And that will give you the error rate, which can make more comparable results. Okay. You're scaling it. It gives a distinct advantage over certain raw error counts. Now, how? Let's look at this example. Okay, I will just clear this. Okay. For example, there is a vitals data set which has 400 rows, and there is demography data set which has 100 rows. Okay. This is the information for the same 20 subjects. Now, in the vitals one, we identified the error as 40 errors were found in 400 rows. And here, 20 errors were found in 100 rows. So that is a simple math. You basically divide it by 40 by 400 and here 20 by 100. Obviously, just by the look of it, it feels that 40 rows vitals is more erroneous. However, when you scale it to 10,000 fields, okay, so if both were 10,000 fields, now here the denominator, here it was 100 and here it was 400. So the comparison is not very easy. So now when I scale it to 10,000, that means 10,000 rows of vital signs, 10,000 rows of demography. Then with the same thing, what would be the error rate in 10,000 fields? When you do the math, it comes out to be 1,000 in vitals, but 2,000 in demography. So just by looking at this initial number, it was giving us a false sense that demography is more clean than vitals. But when the denomination of both the data sets were the same, you realize that in per 10,000 field, error rate of demography was higher. So this is a similar math-like percentage. When a percentage is being used for a comparison, it gives better understanding of the actual situation. So, like we said, it is twice that of the data sets, even though the records were less. So, in spite of less records, the errors were more. This can be compared. So, obviously, you can compare different kind of data sets using the scaling method. And you can also compare the data across even trials, that vitals of trial A versus vitals of trial B. Which trial has more issues in the vitals form? That comparison is also possible. Okay, so this error rate, using the error rate is a common scale of measurement for data quality. So, acceptable quality. While using the error rate, remember that it is just a part of the data quality process. It is important to know if even if the errors are even critical or not, and if they have occurred on the critical or non-critical field. So this is very critical, very, very important line here. Okay. Uh, it is absolutely essential to know that whether the data has occurred on critical or non-critical field. If the error rate is because of critical field, then definitely there is some problem with the way data has been collected. But if it is on a non-critical field, we can make an informed decision if the errors on the non-critical are really going to affect the data in any way or not. If the high error rate is on the non-critical field, they will have very little impact. And then the organization can even determine if it's really worth the time to clean such a non-critical data error. So knowledge of the error rates will help you choose the process paths, how to process the data, even the technologies that can be used so that your quality improves and quality is good for the organization. In the absence of the industry-wide standard for acceptable error rates for CRF to database quality control, quality data means different things to different organizations. So popular definition of an acceptable quality level includes rates of 50 errors per 10,000 field overall, or different standards for critical and non-critical. Okay. So usually they are saying that acceptable rate is you have overall quality is 50 errors per 10,000 fields. So your study database on a whole is good. But if you divide it to non-critical and critical variable, then for critical variable expectation is good quality data is between 0 to 10%. That's like an acceptable industry norm. And for non-critical variable, it could be from 20 to 100 errors per 10,000 fields. So while calculating the error rate, it is important that the units in the numerator and denominator are of the same comparable results. Following data issues, if we are not considering them, it could result in the units or the results getting skewed. For example, the data errors which are involving derived fields, should we even count derived field errors? Because it's the central designing issue. So derived fields should ideally not be counted as a personal issue or the personnel issue. Is an error in the month and year field of a derived date one or two issues? So if there is a date field and both month is wrong and date is wrong, but for another issue only month is wrong and date year is correct. So how do we classify? Should both the instances be counted as one issue, or month error should be calculated as a separate issue, year issue should be calculated as a separate issue? How should the errors be counted in headers that are entered one time, then electronically populated throughout the study pages? So this is header issues is uh, uh, basically whether when manually every time we are entering the data, or when it is centrally entered wrong once, so the same wrong value gets populated throughout the rest of the pages also. Then should all the rest of the pages have the same kind of errors? Another example could be if there was a check which was misfiring within the clinical database. And because of that, the actions that were taken by the team was generally uh, to close those queries, irrespective of the data update. So in that case, should all those queries which have been closed by the study team irrespective of whatever was the data response, should it be considered as one error, or it should be considered the number of times or the number of misfiring checks that were closed? So all these considerations have to be made before arriving at the final error rate. With this, we come to an end to the session. Here are certain references. There's also a very good article, which I would definitely ask all of you all to read through. Thank you.